Now we're moving on to lymphocytes. This is drawing 41, and it should say leukocytes part 4, lymphocytes, in parentheses, B cells. So first I'd like to point out that the difference between the word leukocyte and lymphocyte, these are really easy to get confused, and I think I used to confuse them until I really started doing these drawings and got it straight in my head. Leukocyte means all white cells, any kind of white cell. Lymphocyte means just the cells that came from that lymphoid stem cell. Remember on the right hand of our chart, right hand side there was the lymphoid, and over on the left there was the myeloid. So these are all the cells that come from that lymphoid stem cell, and they would be the B cells, the T cells, and the NK cells. So those are the three types of lymphocytes. Now we're going to look at the B cells first. Okay, so B cells start in the bone marrow. They're born in the bone marrow, and they stay in the bone marrow while they're growing up. So let's draw a piece of bone right here. It can be really ecky looking, doesn't matter, just like that's fine. Okay, and then we'll draw a little section here that represents the marrow at the end, and we'll put little cells in it. Now of course these are out of scale, you're not going to see, these are like the size of peas if this were a leg bone, but we're not going to worry about scale right now. So the B cells stay in the bone marrow until they mature, then they migrate to lymph nodes. Also, spleen and tonsils. The spleen's like a giant lymph node, and the tonsils are kind of sort of a type of lymph node. Part of the maturation process is to learn to make one particular type of antibody. Antibodies are also called immunoglobulins. So you can use the word antibody or immunoglobulin interchangeably. So I'm allowed to talk about one, I can say antibody, and 1 second I can switch and say immunoglobulins or IG, and it's the same thing. Okay, so I'll probably do that sometimes. I'll probably switch back and forth without even thinking. So if I say IG or antibody or immunoglobulin, same thing. I know that's confusing. Why do they have to do that? But that's just the way it is, so I'm going to try to get you used to it. Okay, so we know where they come from, and the important thing is they stay here to mature. We'll see that's not true with the T cells. So that's an important point here, and I'm going to focus in right on this little part here, and I want to draw this is our kind of attempt at a realistic cell. Now we didn't color the macrophage. We didn't really put any color on that one. We'll do some color on this one. So lymphocytes have a very large nucleus. Looks something like this. Let me show you. Here is a picture of a lymphocyte. Once again, these are red cells. There. So they're a little bit bigger than red cells. And that's the nucleus. So this is the rest of the cell. This is all the cytoplasm. Out here would be endoplasmic reticulum, the Golgi bodies, the lysosomes, everything else has only that much space. And they get mature. Here's one that started to be active. That was an inactive one. It's kind of a baby one. When they start to mature, there's more cytoplasm. The cell gets a little bit bigger, and you can see there's some organelles showing up now. It's a plasma cell. We'll talk about that in a minute. And there's another active B cell. There, you can see the plasma, or the cytoplasm area is a little bit bigger now. And here's a picture that has a whole bunch of them. Now, this is not really a good thing. You don't want quite that many of these guys. If a professional pathologist looks at this slide, they're going to say, something's wrong here. Okay, but the slide is nice because it shows us lots and lots of these active B cells. So it's kind of pretty, isn't it? It looks like a kind of artwork, like a batik design or something. All the different pinks and purples and blues. It kind of looks pretty. Okay, so that's what they really look like. So our approximation is going to be, going to make this nucleus kind of a dark blue or purplish color, and then we'll make the outside kind of a light blue. Now remember, once again, I've said this before, I'm going to say it again, real cells don't have this color. These are stained. Okay, so that bears repeating. Now this is a B cell. We're calling it a B cell here, but it would look the same if it was a T cell or an NK cell. All the lymphocytes pretty much look the same. You really can't tell whether it's a B or T just by looking. When it starts to mature, like as the B cell starts to differentiate and become active, then it looks a little more, they're a little more distinctive. But inactive B and T cells look the same. Okay, so let's put an arrow here pointing at the DNA, the nucleus filled with DNA. The DNA in the nucleus will direct the production of one kind of antibody. Antibodies are super small, too small to see with a regular microscope. They're the size of enzymes. I mean, they're tiny, they're just molecular. All right, so you're not going to see any on here. We'll just put here antibodies too small to see. So in our realistic picture, I'm not going to draw the antibodies. Now we're going to draw some cartoon pictures where the antibodies are going to be these giant Y's, okay? But we're going to leave our realistic picture without the antibodies so that you understand you can't see them. So this is also kind of 10 to 12 microns. We're kind of keeping track of how big everything is. Same thing, all these little white cells, basically the same size, a little bigger than a red blood cell. All right, so that's it. That's all we have to do on that one. So let's go over here and look at this, pick up this theme here in the antibodies, immunoglobulins, and look at them a little more closely. Okay, so we're going to draw an antibody, and you're going to keep it in the center here. Something like this. I'm going to draw it in two halves, like that. And then the other half of the Y like this. And then there's one more little piece right under here and right under here. That's it. Now these big pieces and little pieces have names. The big pieces are identical to each other, and the little pieces are exactly the same. The big ones are called heavy chains. Now, why would they be called chains? Well, you think about it, a protein, because they're made out of protein, a protein is a long chain of amino acids all folded up. So this one is bigger, so it's going to be heavier. And so if this is the heavy one, obviously, right, this will be the light one. So they're called the heavy chains and the light chains. And again, they're exactly the same. These little pieces are identical, and the big pieces are identical to each other. So it's like symmetric. Same thing. Okay, now on the end here, you don't have to draw it differently, but if you want to put a little chip out of it, so it looks like a little circle could fit down in there. This is the antigen-binding site. Remember, we've been seeing those little orange circles sticking? Well, if you want to, you can put one in here. They're not going to be perfect circles, but in real life. But okay, so this is antigen. And the little hole, we'll put an antigen here, and I'm going to leave mine blank on this side. I call it the Antigen-Binding Site, S-I-T-E, right, for meaning location. It's called the Antigen-Binding Site. And this section here, this top part, I'm going to put one of those little bracket thingies up here. That part here is called the variable region, meaning it can change, it can vary. In fact, it varies a lot from B cell to B cell. Every B cell has a tip, a variable region that's different from every other B cell. They're all unique. And the process of how that happens in the DNA is pretty amazing. This is the one place where your body allows mutations. All the mechanisms in your body are basically set to prevent mutations. Your cells are set against mutations. That's something that you really need to remember. Also, when you think about, is evolution possible? It's built on mutations, and they talk about all these mutations. You do need to remember that cells are absolutely set against, they do everything they can to prevent mutations. They have spell checkers and editors, they have recycling, they go along, they check if a protein is folded badly, they get rid of it. There is a huge amount of supervision going on when things are replicating. So there's lots of safety designs in it. But here, this is one place where there's no safety mechanism, and the genes, the DNA gets shuffled around, and so amazingly, so that you get these crazy combinations, random combinations of shapes, and that's important because your body doesn't know what kind of shape is going to be coming in. The pathogens could have, who knows, there could be some species of worm your body's never seen before, and will it have the right shape? And if it doesn't, it'll have something, maybe it's close, and then the body has a system. If it's close, the B cell can, the one that's closest can adjust that design. It can be, okay, that's almost it, and then it can adjust it a little more and get closer and closer and closer to the right shape. It's really amazing. That's a little bit out of the scope of this drawing here, but I just had to mention it. It's just a fascinating process. Okay, so let's write here that this is this part, meaning the variable region, this part is a unique shape for each B cell. And let's add, shapes are random. There's no planning for pathogens here. It's just random. And we had made a note down here that our bodies make 10 million different shapes. Most will never be used. What are the odds? You got 10 million different shapes. What are the odds that one of them is going to be the exact right match for a pathogen? Well, 10 million, you might have a pretty good shot, okay? Or at least get close. But it takes that many different random variations to cover all your bases. So most of these B cells will never be used, and they'll just die and get recycled. They'll get eaten. They'll either kill themselves or they'll get killed by a macrophage. And who's the cleanup crew? The macrophage. Okay, so let's go down here and draw some cartoon B cells. This is going to be fun. Okay, there we go. So when the B cells go mature, they go to the lymph nodes to sit and wait until they are needed. These cells are called naive because they don't know what to do yet. Naive isn't really a negative word. It doesn't mean stupid. It just means you don't have any experience. You just don't know enough yet. So they're mature, but they're not active. They don't have a job yet, so they're naive. So let's draw a naive B cell down here. We're going to draw 3 of these guys, so we'll leave space down below. Draw a head, and then because it has that big nucleus, we could just kind of go like this. We'll give him a rim. He's going to look like he's in a space helmet because of that, but it'll be all right. And then the T cells we're going to see have like they have like a T down below, so it looks like they have arms. So we'll give the B cells a B body. They're going to look like snowmen. Give them some stick arms. Okay, so there's his letter B, there's his head, and then his eyes, little circles that are looking up, and just a little dot mouth. Okay, doesn't he look like doesn't know what to do? He's clueless here. I don't know, what do I do? I have no idea. Who am I? And let's give him stick arms to remind us that he makes antibodies. And we're going to call this naive, and actually there's 2 dots over the I, which tells you pronounce it. Naive. Naive B cell. And let's point to these and say capable of making this particular antibody. But it is not making them yet. Okay, the body's very efficient. Why make whole scads of antibodies that you're not going to need? I mean, if all 10 million of these things started cranking out antibodies all the time, your bloodstream would just be full of antibodies, and that would be no good. So he's going to wait until he gets instructions. He knows how to make them. He's showing them. He's like, I can do this if anybody wants me to, but I'm going to wait and see if I get instructions to make more. Okay, so underneath here, we're going to pretend he gets activated, and we'll see how that happens in a few drawings from now. When activated, they become, okay, either, so this is like a divide. Remember, it's sort of like the monocyte deciding what to do. Okay, it can turn into let's see, we're going to make these guys look the same. Except the expression on this guy's face now, we're going to give him a big smile because he knows he has a mission in life now. He knows what to do. He's got a job. Give his arms. And his job is going to be, okay, make a bunch of those antibodies. Just start cranking them out here. So he's got a lot on himself now, but they also float off. They go floating off and into the bloodstream to be delivered to where they're needed. And he is now called a plasma cell. I suppose because he's putting antibodies into the blood plasma. Okay, maybe? Or he's floating around the plasma doing this. But he's called a plasma cell. And he cranks out IGs. See there, I'm going to use IG instead of antibody. Exclamation point. He's cranking out. He's going to, in fact, he's going to burn himself out. He's not going to live very long. He's just going to go out in style, cranking out antibodies, use up all his cellular energy making these things. On the other hand, it could also become same body. Now his face is going to be different. He's going to kind of look like he's asleep. He's still got a big smile because he's got a job, he's got a mission. His mission is to sit here and we'll put a little thought bubble. And it says, I remember. He's going to sit and think. It's like that story, if you ever read the story about the little Frederick the mouse that sits there and thinks and remembers about what it was like in the summer, and that's his job in the community. He's to remember. Well, anyways, okay, so it's kind of like that. So he is given the job of remembering that he was needed, that this one was necessary, needed. Remember, okay, remember, we need to do it one time, so he's called a memory cell. And he remembers what was needed. So if it ever is needed again, he's ready to go. So his deal is that he lives, sorry about that. His deal is that he lives a long time. In fact, he can live for years and years, possibly even your whole life. You know how once you get chickenpox, you never get it again, or once you get some of these viruses, they say you never catch the same thing twice. Well, that's thanks to these memory cells. Because the next time the pathogen, they say you won't get chickenpox again, but that doesn't mean you won't get exposed to it again. You might meet the virus again, but this time your memory cells are like, I've seen that one before. I know how to defeat it. And your body will just take care of it, and you'll never actually feel sick. It'll fight it off right away, thanks to this memory cell. And that's also how vaccines work. You can give your body, stimulate your body to make these memory cells, even just with the samples in the vaccine, so that your body remembers how to fight it. Okay, so let's go over here. As a little reminder, what do antibodies do? Stick to antigens. What is an antigen? Anything not self. That's just our quickie reminder. And let's see, right here, let's just draw a couple things. What do they stick to? Well, you could draw a little Y here. Two little, yeah, it's got a little, well, wait a minute, what is not self? I don't know, what's not self? Now, pollens in the air, you know, they're not self, and maybe, I don't know, from stuffed dander from your cat or dog, if you're allergic to that, mold spores, maybe, or tiny things. Okay. And also viruses, right? All kinds of things, anything that enters your body. There's a different kind of antibody. Now, some of them just clear them out. The ones that stick to pollen, sometimes they cause allergies, but that's because the stick here, the sticks can be actually put up here, actually. Let's put up here. The stick, the base, what do you call the base here? The base can be one of 5 types, and they are named with letters. It can be IgA, IgE, also there's IgD, which you don't hear too much about, IgM, and IgG. So the letters are A, D, E, M, and G. And they all have slightly different functions. And the ones, remember we met about the IgEs, the ones that cause allergies? Well, the stick, we have A sticks, B sticks. They think that maybe what the deal is with allergies is that the IgAs don't really cause any problem. They don't cause anything. They're there to just, they're in your mucus. A lot of them are in the mucus. So they're there to just stick to pollen grains and things and help to wash them out. So this section here doesn't cause the problem. Okay, it sticks to the pathogen. If this IgA, if the stick is A, then the pathogen, it doesn't stick to body cells. The IgE goes and it sticks to those mast cells, right? That's not so good. So your body can decide which kind of stick to put on it. And if it suddenly decides, you know, that it wants to switch, it's called class switching, it wants to switch, and it starts wanting to put a different kind of stick on it, it can do that. And I experienced that. I used to be allergic to a lot of different things, and now I'm not anymore. And I think it's my body no longer puts the E stick on, it's now putting the A. I'm not allergic to cats anymore. So how can you be not allergic to cats? Well, if my body decides to make this the A's, then I don't have a problem. They don't stick to my mast cells anymore. So just a little extra tidbit about the antibodies. All right, so let's go down here and finish this up. What do antibodies do? Let's put a picture here of what they do. They cover pathogens. So let's make our little guy, remember the little guy that got stuck in the phagosome here? All right, well, this is an alternative fate. This is another thing that could have happened to him. He could have been covered, let's put a face on him. He's going, ah. He could have been covered with antibodies. Covered. Now remember, the Y end, the fork end, this end, right, sticks to the antigens. And the base of it is what sticks to your body cells. So when the antibodies stick to a pathogen, that fork end of the Y is what sticks, right? You wouldn't get this. You wouldn't get that. No, that's what happens on mast cells. So make sure you get your Y's going the right way. So some would even stick to its flagella down here. You'd have antibodies to this. And each one of these antibodies is sensing a protein, a section of proteins, a little pattern, right? So, remember the plasma membranes? And it looked like a sea of ping pong balls, and there's all kinds of little things. Actually, I have a picture here to show you an outside of a cell. Here's a computer-generated image somebody did of the outside of a cell. Now these are greatly exaggerated, but just to show you the idea of there's all these different receptors and protein things and shapes and everything on the outside of a cell. It's kind of the way a cell sees it. So some of these antibodies are going to stick to, maybe there's, this section. It's like as if they're different colors or something. And there's different shapes. The flagella is going to have different protein patterns. So the antibodies that are sticking here are not the same ones that are sticking here. Okay, so we could make them, if we'd made them different colors, that's a blue one. They're all antibodies, but they're different types. And so they'd be made by different B cells, wouldn't they? Every B cell makes only one kind. So it takes a lot of antibodies from a lot of B cells to get this guy. Maybe the ones that stick to the tail down here are green. And they can test for this, when you get sick and they want to find out what you're infected with. This is what they're testing for a lot of times. They're testing for what kind of antibodies your body is making. They're actually testing for the antibodies, okay? Well, actually, I shouldn't have made those green because I used green down there. I'll rub over them with pink. So lots of different types of antibodies sticking here. And this guy is he's saying what? Quote, something. I can't breathe. Okay, so he can't move. You're choking him out. And of course, this would be three-dimensional. They'd be stuck everywhere to his body. Right? So that's one thing it can do. And then the pathogen, he really kind of wanted to attack your body cells and make you sick, but it's a little bit hard when you're covered with antibodies. So that kind of puts him out of commission. So that's one thing it can do. Or it can do this, or it could say and. Maybe I should say and, because it could do both. And we'll make some round guys here. These are what, a cocci. Round bacteria. This guy would be a bacillus with a flagellum. Okay, and they're sad because they are all going to get stuck together. And so they've got these. We could forget the sticks. It's too much work to draw sticks if you just draw little V's for the antibodies. So they actually cause the cells to stick together in clumps. It's like chaining them to each other. It's like, well, they like each other, but they didn't really want to be stuck to each other. Can you imagine if you took 10 people and duct taped them together? How well could they do anything? Not very well. So they can't move, they can't do anything. And so what's going to happen to them? Well, macrophage is going to come along and gobble them up, right? Yeah, pretty much. So they can, or make them stick together. And the picture says it all, but caption just says they're stuck together. Stuck pathogens. Okay, one last thing. Antibodies can function as little flags. Then right here, one of their job, what they're doing is they're like little tags that say "Eat me." When a macrophage or a neutrophil comes along, a dendritic cell, or we'll find out like there's also killer cells. So it's like a tag that says "Eat me" or "Kill me." I could add here, or "Kill me." But for the macrophage, since we're talking about that, let's draw a big macrophage down here. Ooh, and he's got a open mouth. Looks like a big fish or something coming in. It's supposed to be a macrophage. Macrophage. There's little lysosomes. It looks like some kind of a spotted tropical fish, I guess. And he's thinking, yummy IGs. He likes to eat. He's attracted to those tags. So here's our poor little bacteria down here. He's like, uh-oh. He is covered with these things, and he knows the macrophage likes to eat them. And oops. It's like being smeared with bacon grease, and then they let the wild dogs or the wolves out. Okay? He feels like a sheep that's smeared with bacon grease, and this is a big wolf coming to eat. Not a good situation. Okay, so there's a pack here. Pack of hunting neutrophils, right? I'll put a letter N so we know what these guys are. Like little Pac-Man or something. So here's his little... We'll draw one more here coming in from the side. I'll put down here, hungry neutrophil army. And that's not what the bacteria wants to come. And they're attracted to these IGs. And so he's thinking, I'm doomed. All right, so very silly drawing, but it makes the point, doesn't it? These guys like to see, you could put a sight line. Oh, yeah, I loves. He's thinking the same thing. Yummy IGs. They want to eat anything that's covered these tags. They want to eat them. So that's the other job the antibodies have.